Greetings everybody. Today's class is about digestion and absorption of lipids. And I am Dr. Mudassar who is going to speak few words about it. So let's begin. What is digestion? It is a process by which the biochemical transformation of complex large food particles which cannot be easily uh, absorbed are broken down into simpler forms which are suitable for absorption and assimilation enzymatically. So this digestion has got few salient features. They are: this digestion is done by special chemicals called as enzymes by a process of hydrolysis. And one more thing we should know, that not one digestive juice does not possess all the enzymes necessary for digestion. There are different types of digestive juice which are secreted right from oral cavity till the intestines. Next important salient feature of what occurs in digestion is there is simultaneous absorption of whatever is digested or broken down into smaller particles, which is very essential for assimilation. The four types of digestive juice are saliva, salivary juice, gastric juice, pancreatic juice, succus entericus, and bile. What is this absorption? Absorption is a process by which the end products of digestion which is breaking down of complex food particles into simpler food particles is passed through the intestinal epithelium to enter the bloodstream, where it is redistributed into the milieu interior, or it is contributed to the homeostasis of our body. Now coming to the topic proper of today's class, that is digestion and absorption of fats. Before going to the digestion and absorption of fats, let us see what are the different types of fats present in our diet. They are basically classified into three types. The simple fats, example triglycerides and cholesterol; compound fats or complex fats, which are phospholipids; and associated fats, which are seen in steroids and soluble vitamins. The source of our dietary fats is both from vegetables and animals. For example, vegetable source is the vegetable oil what we take daily in our day-to-day life, for what we consume in our day-to-day life. That is the dietary fat from vegetable source. And from animal source, the common fat we take is paneer, ghee, butter. These are the dietary fat from the animal source. This daily dietary intake also varies from person to person, and from person to person which widely varies between 25 grams to 160 grams per day, situation to situation. Like during festivals, during celebration, we take a lot of fat. Why do we take fat? Fat makes our food palatable. You might have seen a lot of fries are served during festival. It's a way of celebration, and that's how fat make our food more palatable. We eat, we celebrate. We celebrate by eating, and while eating, we celebrate. That's our culture. So next is the site of digestion of lipids. The main site for digestion of lipids is mainly intestines. But you should know that there is certain amount of salivary lipases present in the salivary juice, and also gastric lipases present in the gastric juice secretion. But they don't have but they don't play much a role. Why? Because for lipases to act, it requires a pH of more than 4. Now, what is the pH in gastric lumen? It is somewhere around 2 or less than 2, due to presence of high quantity of HCl, because of which these lipases are inactive and they don't play much role in digestion of lipids in those parts of GIT. but the moment food reaches intestines, it is acted by the lipases present in the intestines, and it helps in digestion of lipids. So that is why the main site for digestion of lipids is intestines. Now, what is the mechanism of digestion of fats? It involves three main mechanisms. One is emulsification of fats. The large fat dropules what is present in our food are broken down into small fat globules with the help of bile salts, which is called as emulsification. After they are broken down into small droplets, the surface area of these fat droplets is increased so that the lipolytic enzymes present in the pancreatic juice and intestinal juice can act and cause hydrolysis. After hydrolysis occurs, they are broken down to simpler fatty acids, which is converted into which converts the water insoluble fats into a specialized structure called as micelles, which helps in transportation and also helps in solubilizing or containing the fat in the water, which is insoluble in water. Now, how these process occurs, let us see. First, the emulsification of fat. The pancreatic lipase is the main lipase, and it is mainly a water-soluble enzyme which acts only on the oil-water interface. Now, if a large water droplet, oil droplet is present, the fat droplet is present, then it would take years together to digest it. So to solve this problem of water-soluble pancreatic enzyme, pancreatic lipase, what happens in our body is there is secretion of large amount of bile salts. Now, these bile salts, they are amphiphilic molecules which are having both hydrophobic and hydrophilic ends. So what they do, they penetrate through the neutral fat and break down this neutral fat into small fat globules, and these small fat globules are present in the core of this bile salts which surround these fat globules, small fat globules. While surrounding these fat globules, the hydrophilic end are present to the periphery and hydrophobic ends are present towards the core, which hold and break down the large fat globule into smaller fat droplets. This is done by bile salts. This action of bile salts is called as detergent action. What it is? It is the emulsification of fats which is caused by amphiphilic molecules, bile salts having hydrophilic ends and hydrophobic ends, which the fat globules are broken down into smaller fat lipid molecules which increase the surface area and lowers the surface tension, which increases the surface area for pancreatic lipase, which is a water soluble enzyme, to act on them. This is how, with the help of emulsification, large fat globules are broken down into small fat droplets by the action of amphiphilic bile salts, which increases the surface area for pancreatic lipase to act on it. Next is hydrolysis of these fat droplets by pancreatic and intestinal lipases. Pancreatic lipases are of three types: pancreatic lipases, cholesterol ester hydrolases, and phospholipases. The cholipase is a type of protein present in the pancreatic juice which removes all the bile salts which are present around the fat droplets. After the removal of these fat droplets, the fat is exposed to pancreatic lipases, which convert the fat into triglycerides, then diglycerides and monoglycerides. While the fat is being converted from fat to monoglycerides, two fatty acid molecules are released and one monoglyceride is released. Next is cholesterol ester hydrolysis, which mainly esterifies and hydrolyzes the cholesterol present in the diet. Phospholipases hydrolyzes phospholipids into fatty acids, separates the fatty acids from the phospholipids. Next type of lipases present in our body are intestinal lipolytic enzymes, which are secreted mainly from the brush borders of the epithelial cells. They also have very similar action to pancreatic lipase, which convert the fats into monoglycerides and also release the two fatty acid molecules. This is how, by hydrolysis, fat droplets are converted or broken down into simpler chemical forms, that is monoglycerides and fatty acids. After they are released, remember these fatty acids and monoglycerides, they are highly insoluble in water. They cannot dissolve in water. To solve this problem of insolubility of these fat molecules or lipid molecules, again we have a very interesting, nature has provided a very interesting solution to it. What? That is formation of micelles. What are micelles? Micelles are small water soluble disc shaped or cylindrical particles formed by aid of bile salts. Again, bile salts are amphiphilic molecules with hydrophilic end and hydrophobic end. So each micelle is composed of a central fat globule surrounded by 30 molecules of bile salts in such a way that their lipid soluble or non-polar end, hydrophobic end, are directed towards the central fat globule, and water soluble polar end fan out to form a outer covering of the micelle, like here what you are seeing here. So the monoglycerides, free fatty acids, two free fatty acids which are released from the digestion are quickly incorporated into the central portion of these micelles, and they are transported towards the enterocytes from where the absorption occurs. How does absorption occurs? Absorption mainly occurs in duodenum, and it is completely, the fats are completely absorbed by the time the chyme reaches the mid of jejunum. It involves following steps, that is transportation of micelles towards the brush borders of the membrane, diffusion of lipids across the enterocytes, transport of lipids inside the enterocytes to the interstitial space, and then transport of lipids into the circulation. How it occurs, let us see. The micelles, they contain, as I said, amphiphilic molecules of bile around it. That is removed, and whatever free fatty acids and monoglycerides are there, they enter the lumen by binding to the receptors present on the brush borders of the luminal side of enterocytes. After they enter, small chain fatty acids, they directly diffuse out of the enterocytes. Remaining fatty acids, they again combine with monoglycerides and form triglycerides and also phospholipids. The cholesterol also present inside the enterocyte combines with fatty acids and forms esterified form of cholesterol. All these esterified cholesterol, phospholipids and triglycerides, they combine and fuse to form oil droplets or fat droplets called as chylomicrons. These chylomicrons are excreted out of the enterocytes by a process called as exocytosis. After they are exocytosed, they enter the interstitium. In the interstitium, again, these chylomicron droplets, they coalesce or fuse to form large oil droplets which are absorbed into lacteal, which is a part of lymphatics of the intestines in the gastrointestinal system. From the lacteal, it enters the thoracic duct and then into the blood circulation. That is how the fats what we have taken in our diet, they are broken down into simpler molecules and then packed in micelles. Again, they enter the enterocytes. From there, they form chylomicrons which enter the blood circulation. This is how absorption of fat occurs. Now, the last part of today's class, that is applied aspect. One of the common problem related to lipid digestion is lipid malabsorption, which is quite common when compared to carbohydrate malabsorption and protein malabsorption. The commonest complaint with which the patients who are suffering with lipid malabsorption come are steatorrhea. What is steatorrhea? Increased amount of fat or pale colored passage of pale colored stools is a common manifestation of this fat malabsorption. Generally, parents, they come with their children with a complaint of failure to thrive and also passage of this white color lipid containing stools, which is steatorrhea. What is the cause for this? The most common cause for lipid malabsorption is deficiency of pancreatic lipase, which is very essential for digestion of lipids. And also if there is any deficiency of bile due to disorders of liver, as in case of cirrhosis of liver or any congenital disorder, metabolic disorder, or disorders of gallbladder, lead to lack of or deficiency of bile, which results in lipid malabsorption, for which the commonest complaint is steatorrhea. That's all about digestion and absorption of lipids. Please go through this lecture. If you have got any doubts, please put it in the comment section, and also you have to complete one assignment which I will be giving along with this. Thank you all.